Today we're going to be talking about the broader discipline of biogeography. Biogeography is a rather vast field, and people teach entire courses in biogeography, but we're going to be trying to cover some of the important elements here that influence the contemporary patterns of diversity and distribution that we see today, and understand some of the processes as well. So what is biogeography? Brown and Lomolino define it as the science that attempts to document and understand spatial patterns of over the earth. It is the study of distributions of animals, both past and present, and of related patterns of variation over the earth in the numbers and the kinds of living things. Now, what are the kinds of questions that we typically ask in biogeography? Why is a species or genus confined to its present range? What enables a species to live where it does and not elsewhere? As you can see already, that there's many elements of biogeography that will overlap with both ecology and evolution. What roles would climate, topography, and biotic interactions play in limiting distributions? Questions that we asked when discussing macroecology. How do organisms replace each other along environmental gradients? What are the species' closest relatives and where do they live? How have historical events shaped distribution? And why the fauna and flora of isolated regions so distinctive? And the classic one, which we've addressed repeatedly, why are there more species in the tropics? We talked briefly about Aristotle and his role in classifying living organisms, actually like in classification in general. Aristotle described the natural history of more than 500 species of animals, and he was classifying them on the basis of what he called 5 predicables. Species, which was a specific category; genus, a more general category, and what differentiated them from each other, what properties they had, and what he called accident. And he divided them on the basis of the principle of logical division. Plants and animals in the scheme were defined by genus and species and differentia. And it's always useful to acknowledge Aristotle, because a lot was the Western science that developed in the 17th and 18th century was really based on some of the work that he'd done more than 2,000 years before. While we credit the discovery of evolution to Charles Darwin and Alfred Russel Wallace, and the people that came before, geologists such as Charles Lyell, Louis Agassiz, and many other naturalists of the time, such as Herschel and others. There are some ecologists, naturalists, biologists of the mid 19th century that particularly made contributions to what we think of today as the field of biogeography. One of these was Philip Sclater, who described over 1,000 species, 130 genera, a couple of families of birds, and his classic work was on the general distribution of members of the class Aves. And Sclater first described these regions as we know them today. And as you can see in this illustration, he had these are actually his divisions of the different oceanic regions. So while Sclater made these remarkable contributions to biogeography, the other person that's considered as the father of zoogeography in a sense is Alfred Wallace, who I call the prisoner of parentheses, because everybody talks about the discovery of the theory of natural selection as framed by Darwin, and then you always say in brackets and you say and Wallace. Wallace actually was remarkable in many ways because he did not come from a wealthy family, spent his life savings to finance his voyage to the Amazon, and a second voyage to the Malay Archipelago. And in the classic story about about the theory of natural selection. He wrote to Darwin and asked that his paper be forwarded to Charles Lyell, the geologist. Darwin's response to this was that if Wallace had read my sketch written in 1842, he could not have made a better short abstract, which meant that Wallace had come up with the identical idea, and if Wallace had published independently, Darwin would have been scooped. But as it so happened, Wallace had written to Darwin, and so Lyell and Joseph Hooker arranged for Darwin to present both their papers simultaneously to the Linnean Society, and therefore they're co-credited for this work. Wallace is, as I mentioned, often considered the father of zoogeography. His works, the Malay Archipelago, the geographical distribution of animals, island life, are some of the most significant contributions of the 19th century to our knowledge of biogeography. And he developed this detailed map of the world's biogeographic regions, and is also well known for this sharp faunal gap between Bali and Lombok and Australasia that is still known as Wallace's line and corresponds closely to the continental plates. So with that slight historical background, let's get back to our exploration of biogeography, and let's pause and look at the conceptual framework that we developed and tried to understand during our exploration of macroecology. And so here we're focusing on species geographic range, not so much richness, and the factors that we're interested in are less the ecological factors and more the historical factors such as origins, evolutionary origins, and evolutionary dispersal as how species got to particular locations over evolutionary time. This of course is a combination of spatial and temporal processes and involves both where continents were, the routes that animals had to take for dispersal, and the time periods that they took place in. But in addition to the spatial processes of dispersal, there's also diversification or evolutionary change over time. Now, we talk about the evolutionary post-processes as the origin of new species from pre-existing ones. There are both adaptive as well as non-adaptive radiations, and we'll spend some time trying to understand both of them. We'll also examine different kinds of dispersal. Dispersal also is not sort of just one type over evolutionary time. Species can expand through range expansion, jump dispersal, and so on. Let's start with evolutionary processes. And broadly speaking, understanding evolutionary history or inferring evolutionary history is trying to understand how organisms are related to each other, which is also related to the classification of these organisms. So the classification of organisms, which of course was classically done using morphological features, is really an exploration of how these animals are related to each other. Things that are more closely related are classed into certain groups. A great example of this is the classification of reptiles. And traditionally, historically, and to a great extent perhaps in schools even now, crocodilians, crocs and alligators, tuataras, squamates, which are snakes and lizards, and turtles are traditionally referred to as reptiles. So most standard textbooks will say reptilia, aves, and mammalia, right? And these are the groups that they've been classified into. However, if we look at relatedness on a phylogenetic tree, it's long been known even from osteology and morphology that birds are actually quite closely related to crocodiles, and they're really more closely related to dinosaurs than they are to other extant reptiles. And of course, genetic data very strongly supports it. And what this sort of tells us, in a sense, that there's really, from an evolutionary perspective, there's what we currently call reptiles is not a is not a viable group. And we could either, if we wanted to retain sort of crocodilians within the group of reptiles, then we'd actually have to include birds as well in this group that we call reptiles, or we'd have to restrict the evolutionary grouping or classification of reptiles to just in this particular tree to crocodilians, to tuataras and squamates, and turtles and are a separate group. Birds and crocodiles are a separate group. We use evolutionary biology and more contemporarily phylogenetic methods to understand relatedness, and this guides our classification of those groups. Now, one of the reasons why organisms also have morphologies that resemble one another is that there can be convergence across groups, which is when unrelated organisms evolve structures, traits, or morphological features that have the same function, such as this particular shape that we see across fish, reptiles such as ichthyosaurs, puffins, which are birds, as well as cetaceans such as dolphins. And at a broad classificatory level, we don't confuse birds with dolphins or fish, but at a finer level, often a species may have morphological features that evolved through convergence and may mislead attempts at classification. The other broad process that we're more familiar with is that of divergence, as in the classic case of Darwin's finches, where in response to environmental differences and along with geographical barriers, species can, in a short period of time, evolve into multiple different species that are adapted to different habitats. On the subject of classification and relatedness, if we look at apes, we are most closely related to chimpanzees, and then on to gorillas, and further on to orangutans. And this, when first proposed and eventually supported by evidence from genetic data, may have come as a surprise to some. So while the broader exploration of evolution is examining the relatedness of organisms to each other, we're particularly interested in the processes that bring these about, right? The processes of speciation and extinction to begin with. There are four broad types of speciation: allopatric and sympatric to begin with are the most significant contrasts. There's also peripatric and parapatric modes of speciation, which we'll come back to in a few minutes. Now, allopatric speciation is broadly speaking geographical speciation, or the divergence of species when there's a separating barrier. Sympatric speciation is speciation at the same location. Parapatric is across environmental gradients. Allopatric speciation itself can be of two different types. One is through vicariant events, which is barriers to dispersal in an existing population. So you have an existing population of animals over here. There was some sort of barrier, either rising sea levels, continental drift, mountains came up, and these usually isolate fairly large populations. Eventually they become different species. You can also get allopatric speciation through dispersal or founder events, the movement of small individuals across an existing barrier. This usually involves a small initial population. Once populations re-establish contact over a period of time, these individuals may start going back and forth again because they've figured out how to get past the barrier, or the barrier disappeared. They may not interbreed at all, or they may produce fertile offspring and re-establishing gene flow. They could also produce infertile offspring, of course. Now, if they interbreed freely and extensively, that gene flow will eventually lead to to these becoming, well, let's not call these different species. Let's say that these were evolutionarily divergent lineages with the potential to become different species. If they start interbreeding freely and extensively again, then the speciation event does not occur. There are other mechanisms. Hybrids may be less fit, so they may interbreed, but the hybrids may not survive, and these mechanisms might lead eventually to their complete divergence. Barriers may be in the form of mountains, as in the graphic of frogs that I showed, or in the form of land masses that divide oceans. One classic example is the Isthmus of Panama, which formed about 3.5 million years ago, and this shows the difference, the linear divergence of Atlantic and Pacific pokefish. And so after the formation of the Isthmus of Panama, the barrier to this dispersal then led to these diverging into separate species. And this has affected many species that were found in the Pacific and Atlantic sides of the isthmus, that subsequent to the formation of the isthmus then evolved into different species. An interesting case is that of ring species, where adjacent populations actually have gene flow between them, but as you move from one population to the next, these no longer have contact with the original populations, and eventually across multiple adjacent populations might meet each other, but fail. But those adjacent populations might have a reproductive barrier. One classic example of these are the circumpolar distribution of gulls, of herring gulls at one end of the spectrum, and Siberian blackback gulls and other lesser blackback gulls and so on. That sort of there's gene flow between 1 and 2, 2 and 3, 3 and 4, 4 and 5, but 1 and 7 are believed to be reproductively isolated from one another. Sympatric speciation occurs where there's the diverging lineages are not separated by geography, and could happen through disruptive selection, chromosomal changes, which is changes in number, aneuploidy or polyploidy, rearrangement of chromosomes, and so on. And some classic examples are from isolated lakes, such as cichlids in Africa, sticklebacks in British Columbia, herrings in Scandinavia, and so on. I touched briefly upon parapatric and peripatric speciation. These are similar to allopatric speciation in the sense that they occur. That in one case there's a founder that's separated. In the other there's a small part of the population at the edge of a larger population that's separated, that first serves as a founder for a new lineage, and parapatric that occurs along a gradient. Here there are barriers to gene flow that are not as explicit or obvious as sort of a mountain or a river or an ocean. The other major process is extinction, which is essentially an ecological process. One must recall the Red Queen hypothesis, that the Red Queen from Alice in Wonderland, which says it takes all the running you can do in the same place. So species have to adapt all the time to biotic and abiotic conditions in order to just stay alive. But it has been found that the probability of species becoming extinct is independent of its evolutionary age. But also that certain taxonomic and ecological groups have higher rates of extinction. So small and herbivorous mammals have been found to have higher rates of extinction than large and carnivorous mammals. And extinction is often driven by stochastic processes, which could be genetic, environmental, or demographic. Extinction in the fossil record is is quite prominent. Most of the species that ever lived on Earth have gone extinct. One of the biggest extinctions was the Permian Triassic extinction, which wiped out most species on Earth about 250 odd million years ago. Of course, there's a very well-documented Cretaceous Tertiary extinction, which resulted in the disappearance of dinosaurs and most of terrestrial and marine animals. The main hypothesis for this is the Alvarez hypothesis, which is the asteroid impact, for which evidence came from predictions of iridium-rich deposits near the Katy boundary. The majority of extinctions occurred actually at this boundary itself. And finally, the discovery of a crater in the Yucatan region of Mexico. Perhaps most recently, the Pleistocene overkill hypothesis, which we'll dwell upon briefly. Apart from the evolutionary processes of speciation and extinction, there is the major biogeographic process of dispersal. I started with Aristotle, and we come back to his work on physics, where he says nature has been defined as a principle of motion and change, and it is the subject of our inquiry. We must therefore see that we understand the meaning of motion, for if it were unknown, the meaning of nature too would be unknown. There's a rather nice quote that he makes about motion and change in general, but I think it applies particularly keenly to our understanding of biogeography. So what is dispersal? Firstly, we use it loosely, and it's often used to allude to an ecological process, which is, for example, movement away from a natal location or where you were born, which can be beneficial in terms of reduced competition with parents and siblings. And this is true for plants, this is true for tigers. Juveniles or offspring or propagules often disperse away from natal locations. Of course, there's a cost to this. As distance increases, habitats become more dissimilar, and your ability to survive may decrease. But importantly, the role of dispersal in biogeography is different from its role as a demographic phenomenon. It's something that affects populations within a couple of lifetimes. Biogeographically, the question that we ask is, does it result in a change in geographic distribution? Now, a juvenile tiger may disperse away from its parent's territory, but it's still dispersing to an area where other tigers are found. So it has not changed the geographical range of tigers. These sorts of dispersal events are often rare and need to be inferred from indirect evidence. But while they are infrequent and stochastic, they are critically important, like beneficial mutations are in evolution. These infrequent stochastic dispersal events are critical for underlie biogeographical processes and result in the patterns that we see. So in order to expand a range, a species must be able to A, travel to a new area, that seems obvious, withstand potentially unfavorable conditions, and then establish viable populations upon arrival, because you may, as a bird, you may be able to fly somewhere, you may think the climate is perfectly fine, but if you are unable to establish a population, if you're a single male or a single female that is not going to lay eggs, then or a tree, that is unable to set seed on its own, then you're not going to establish a new population. There are three broad mechanisms for dispersal: jump dispersal, diffusion, and secular migration. Diffusion of these is the slowest range expansion process. Jump dispersal is the most dramatic, and can come about both due to exceptional dispersal ability on the part of species, but it can also come about due to stochastic events such as storms that might carry propagules of plants and animals across to islands or new areas. Dispersal can be active and depends on the vegility and agility of animals. Birds, bats, large insects, for example, have great dispersal ability. What we know from the migratory ability of birds such as the golden plover that flies all the way from the Arctic to Southern America, Asia, Pacific Islands, travel several thousand kilometers non-stop, and many examples of birds such as Arctic terns and others that are able to make these huge migrations, but also insects such as monarch butterflies and dragonflies, and not as many non-volant animals that can't fly, typically do not have such large migration or dispersal abilities. Of course, with the exception of aquatic animals, whales, sharks, sea turtles are known to make very large-scale migrations. Sea turtles, for example, migrate from their feeding to their breeding grounds. Loggerhead turtles that feed off the west coast of the U.S., of California and of Baja California, are known to migrate all the way across the Pacific to nest in Japan. That's a distance of about 10 to 15,000 kilometers. Leatherback turtles are known to make trans-Pacific and transatlantic migrations. In fact, even the ones that nest in the Andaman Islands have been tracked to all the way to the west coast of Africa, which is more than 10,000 kilometers, as well as a few mammals large mammals that also do make very large-scale migrations, such as elephants. Equally important for biogeographic patterns, mechanisms of dispersal can be passive. It doesn't matter if they're active or passive. All that matters is whether the propagule got there or not. Obviously, many plants that are windblown or plants that are dispersed by animals. Many fungi and microbes. Aerial plankton, for example, can include mites, spiders, and insects. There's also passive dispersal in oceanic systems that I'll touch upon in a moment. Barriers can be physical, as we've seen, and these can be mountains, rivers, oceans. But barriers can also be physiological. The physical barriers are more obvious. Physiological barriers can be the physical tolerance limits of organisms. So this could be temperature, it could be salinity, it could be water availability. So for example, many freshwater fish and amphibians are intolerant of salt water, and since frogs have low tolerance to salinity, in fact, they're generally depopulate on oceanic islands, and so this can strongly influence biogeographic patterns within groups, environmental temperature regimes, and other physiological barriers. There's also ecological barriers such as competition and predation, as well as behavioral and psychological barriers. So there are animals, for example, in the contemporary world, some that are more shy of human settlements, and human settlements therefore pose a barrier to their movement, and others that are actually less shy of human settlements and are able to use that matrix quite effectively. And then there's habitats that the specificity of an animal to a particular habitat can also be an ecological barrier to its movement. So once an animal has moved, it has to establish a colony. Getting there is only half the problem solved. Various things, influence whether a colony can be established. It can be whether the habitat is suitable or not, whether the habitat is patchy or continuous, or whether if it's patchy, then it may be, the probability of establishing a viable population there may be lower, and the propagules themselves may differ. So it depends on whether they have asexual or sexual reproduction. With sexual reproduction, you obviously need enough propagules to produce the next generation. Well, at the minimum, one male and a female. Also, self versus cross fertilization versus apomixis. And finally, the ability of the organisms to reproduce and then survive in that habitat, which again, as previously mentioned, is driven to a great degree by demographic environmental and genetic stochasticity, so demographic being variation in small populations in demographic features such as the survival of offspring, the sex ratio produced, and so on. Environmental changes, stochastic changes that might reduce the survival of individuals, and of course genetic factors such as drift or the accumulation of that might lead to the accumulation of deleterious alleles. I briefly want to sort of give an example of the Western Ghats to demonstrate how we might try and understand this with an actual example of a region. Now, if you look at peninsular India, there's many theories about hypotheses about how animals arrived in India or dispersed out of India, and the dispersal scenarios include, for example, that most of that Indian fauna have Gondwanan origin and dispersed out of India into Asia, and accompanying this is a phylogenetic tree that that reflects this pattern. Alternately, you could have had an out of Asia and into India dispersal of organisms once India collided with Asia. Now, the Western Ghats, again, specifically, we can think about these multiple dispersal scenarios in both directions, both into and out of the Western Ghats, and people have talked about dispersal through the Eastern Ghats, through the Vindhya Satpura, through the Aravalli ranges, and so on. So we can use in contemporary evolutionary methods, we can use genetic data and phylogenetic methods to reconstruct these pathways if we have samples or if we have a sampling of a representation of these taxa from across these regions. Within the Western Ghats itself, one of the really interesting patterns that people have found across groups is that the Palghat Gap, or this across this mountain range, there's a gap here, a valley called the Palghat Gap that goes almost down to sea level. They found that these separate clades or groups of organisms that have been separated for millions of years. So these bush frogs, the northern and the southern clade, the northern that is more grassland dominated, and the southern that's sort of more forest and canopy dominated frogs, separated from each other 15 to 20 million years ago on the two sides of the Western Ghats. A few species went back and forth, but for the most part, these two clades have remained separate from each other. And you might think, yeah, these are frogs, these can't go very far. You actually find that with different species of these Sholakilies, these small forest birds, you actually get divergence across both the Palghat Gap as well as the Shenkota Gap further south. So these barriers in these cases, in the case of the frogs, they divided these evolutionary clades at a much deeper evolutionary time, about 15 million years ago, but they're also resulting in speciation in these Sholakilies at a much shorter, sort of like in a smaller scale of evolutionary time at the species level. That's a Sholakili right there. But you'd imagine that elephants can walk anywhere, and they are, in fact, they do have tremendous ability to cover distances. But despite that, it has been found that elephants as well show at the population level show differences north and south of the Palghat Gap. All right, so let's come back and to our broad framework. And in understanding, the broad geographical range of species, we've talked a little bit about the processes, speciation and extinction that might result in contemporary ranges that we see, and the processes of dispersal, which is a fundamental biogeographic process that also results in the species geographic range. Earlier on, we had examined how summing these up gives us species richness across regions, but looking at the composition, the differences in who is found where gives us an idea about the biogeography of plants and animals. So that concludes our first lecture on biogeography, and we'll come back and look at geological and historical processes on Earth. Thank you.